Very good morning, everyone, colleagues, researchers, students. Welcome to today's Hong Kong IAS Distinguished Lecture Series. I'm Xin Wang, today's facilitator. We are very honored to have Professor Reinhold and Scott with us from Stanford University. So let me just give a very brief introduction about Professor Scott. Professor Scott is Ruth G. and William K. Boyce Professor of the Department of Material Science Engineering, Department of Mechanical Engineering at Stanford University, also at the Department of Surgery at Stanford School of Medicine. So he has been extensively working on integrating new hybrid materials into emerging nanoscience energy applications on the biomechanical functions, the barrier functions of human skins, also soft tissues. So he has won a lot of awards, numerous awards, including IFSCC 2022 Best Applied Research Awards, the Henry Marceau Award from Society of Cosmetic Chemists for fundamental contribution to skin science, etc. So today I think he will share with us with some of his recent work on the structure, function, and biomechanics of human skins. Without further ado, let's join me to welcome Professor Scott.
Well, thank you very much. It's really a pleasure for me to be here today, and I look forward to telling you about some of the work we have underway in the area of human skin. So if I can have the first slide. I thought, though, that I would start, let's see here. Do I need to turn the slides on here, or? There we go. I thought I would first show you where I come from and tell you a little bit about the campus. So this is the front of the university campus, and you're looking here at the original main quad that was built in the late 1800s. And you see there the background of the mountains. So in a way, Stanford looks like a country club where we're playing golf and doing all sorts of interesting things. But in fact, Stanford is really quite a remarkable institution. The campus itself is extremely large. It's over 8,000 acres in size. We have 600 buildings, as you can see here, nearly 50 miles of road. We have our own power plant that generates electricity with a large number of renewable energy sources on campus. We have dams and lakes. We have actually one of the Bay Area's premier shopping centers. Now, I'm not saying that most of the faculty and students can shop there, but it is a significant center. In addition to a number of other features that we have an innovation center, which is shown over here, which supports some of the innovative aspects of startups at Stanford. We have a national accelerator laboratory, so we have a full beamline, high-intensity beamline for studies that's managed by the university, and as well as 2 university and 1 veteran administration hospital. So lots of university hospital and faculty that we're able to interact with. So the picture that I just showed you was in fact just this front here, this front facade of the university. The actual university spans this entire area here. In fact, it goes way up into the mountains and around the other side over there. So very, very large campus. This is the linear accelerator complex that we have over here, and I'll show you a little bit of the work we're doing at the complex for our human skin work. But it's a facility that we use for a large number of studies. If you look more broadly, this is the San Francisco Bay Area here. This is the city, San Francisco. The Golden Gate Bridge is over there. Campus is right here. So we're right on the northern tip of Silicon Valley. And as many of you would know, Stanford has a tremendous involvement and interaction with many entrepreneurial startups and companies. And so we interact widely with companies in this area here. And of course, the Silicon Valley in many ways is now a virtual valley. It's not all just here. And Stanford is very much involved in it. And then coming up here on the East Bay, this is coming up through Hayward, Oakland, and then Berkeley. And apparently there's a public university there that you might have heard about as well. So that's the environment that we work in and where I come from. What I wanted to talk to you about today is work that we've had underway for maybe 25 years now, associated with the structure, the function, and the biomechanics of human skin, with implications in a number of different areas that I'll share with you. But particularly today, wanted to tell you about some recent work involving the sex differences in human skin and how that affects behavior, and also work we've done on understanding topical cosmetic treatments for skin for various applications and their effects on sensorial perception, how we actually perceive our skin. So I don't do any of this work. I just get to travel and tell people about it. The folks that are really doing all of the work that I acknowledge here are many of my PhD students working in this area, as well as former postdocs, master students, and undergraduates that we often have involved in the work, along with collaborations with many different companies, several interactions here in Asia. We have a fairly good new interaction with a large cosmetics company in China, but along with all of these other companies here. This is critical part of what we do, is being able to interact with these entities. So these are the areas that we have been interested in and have worked on. We're interested in aging and what happens to our skin as we age. What are the biological changes that happen? And then what are the potential treatments that can be used as your skin ages? We've been interested in skin damage, particularly exposure to pollutions or to solar UV radiation, and have done a lot of research here looking both at the damage mechanisms as well as the efficacy of treatments like sunscreens. I'm not going to talk about that work today. Several major studies involving the application of topical treatments, and I will say more about this today, and particularly show you some work we've done understanding the mechanism by which you actually have a sensation in your skin and what that mechanism is. This relates to the activation of mechanoreceptors that are actually embedded in the skin, and the first major mechanoreceptors really emerging at the basement membrane, the connection between the epidermal and the underlying dermal layer. We've also been interested in scar formation, hypertrophic scarring, and wounding, and I thought I would show you just a little bit about some of the work we've done in the area of hypertrophic scarring. This is work I did with a colleague, Jeff Gertner, in surgery at Stanford, where we were interested in the mechanoresponsive nature of hypertrophic scarring. It turns out that the triggering mechanism for the formation of hypertrophic scars, and mammalian species like humans scar and form these scars very rapidly. There's a biological incentive to actually heal wounds quickly, but that often results in the formation of a large hypertrophic scar, shown in these images over here. And the mechanisms for this are really complex. There's many underlying mechanisms that are activated that involve gene expression, protein expression, and this forms part of a very complex wound healing process. What we wanted to do was, since many of these mechanisms are mechanoresponsive, we wanted to try and control the environment in which these scars were forming. And what we did eventually was to use a very simple class 1 device where we would shield the wound from the normal physiological stresses that we have in our skin. And we did that with this very simple device that we initially fabricated, which would be pre-deployed and then adhered to the skin and contract in a controlled fashion so that the healing wound really is shielded from both endogenous and exogenous physiological strains and stresses. And it turns out this has a major effect on reducing scar formation. We showed that initially in a porcine model. We then filed a patent around that, which ended up being a very, very strong patent. Surprisingly, this kind of approach to actually reduce scarring had not been shown before. And we then started a company, which is a typical process that might happen at Stanford, called Neodyne Biosciences. We then made some more commercial-looking devices. This is one of the current devices that are available, and ran several major clinical trials. And you can see in many of these procedures here a control side where we use standard of care and the typical kind of hypertrophic scarring that might form, and then a side that was treated with one of these devices that controlled the stress state. Here's an example of an abdominoplasty, and this is one side of the abdominoplasty, and this is the other side that was treated. So it's kind of a nice model to use because you have a built-in control. And you can see this very significant reduction in scarring that we were able to obtain. So this was a big area of study. It went on for many years. The company is still around and is doing quite well now, looking in towards other applications of this technology. Another area that we became very interested in was related to the damage that happens to human skin. This is not a significant damage that might actually be life-threatening. This is the kind of avulsions and abrasions that occur in skin, where it turned out in the literature, in the clinical literature, very little was known about this type of damage, cutaneous damage, so non-lethal cutaneous damage. And so you can see in these high-speed images here an example of some of the studies that we undertook. This is a ball bearing that was basically shot at the skin at different angles and different velocities. And you could see in that image, I'll go back and show that again, the kind of damage that occurs. This is the epidermal layer here being stripped off the underlying dermis. To understand this better, we actually developed a model which involved a dynamic three-dimensional model of human skin with all of the different layers incorporated, in which we were able to simulate with high degrees of precision the exact kind of damage that we were observing. And again, of course, then followed a large number of studies. This is all work on human skin, where we could quantify the type of damage that would result from different kinds of abrasive or dynamic impact. And we came up with a wound damage index, which can now be used to actually quantify these wounds. So those were two areas of study that we've been interested in, involving the full thickness skin and aspects of either scar formation or wounding. I wanted to turn now and say a little bit more about the effects of cosmetic treatments. So skin has a hierarchical structure, and the nature of the individual layers of skin, and shown over here are the thicknesses of the upper epidermal layer, sort of 50 microns to about 1.5 millimeters. The dermis about just over 0.5 millimeter to 4 millimeters, and the hypodermis underneath. Each one of these layers has different evolved properties that is very important in terms of the function of skin. So just a few of those written out over here. The top epidermal layer is known for very high cell proliferation. So there's a renewal of this top epidermal layer, particularly the very top stratum corneum layer, involving the cornification of cells and the subsequent disclamation as these cells are actually released from the top surface. The underlying dermal layer is important in terms of its ability to actually tolerate deformation. So it needs to be strong, it needs to be able to deform without damage. It's also the vascularized portion of skin and also has the mechanoresponsive cells and afferent nerves that allow us to have the sensation of skin, both of touch, tactile sensation, and other aspects of skin. And then, of course, the underlying fatty layers that are important for things like shock resistance and energy storage. So when you're thinking of cosmetic formulations, one of the most important layers of human skin is the very top stratum corneum layer. So this is a layer typically about 10 to 30 microns in thickness, and it's formed of these pancake-like cells in this sort of early model that was described by Marx as early as 1983, where they called this sort of a brick and mortar model. So these cornified cells are held together with small protein connections called corneodesmosomes, as well as these lipid layers between the cells. So what's critical when you actually apply a cosmetic formulation is understanding how that formulation interacts with this particular structure. So it's been a layer of the tissue that we've been very interested in, and dating back to the early 2000s, began to look at isolated layers of the stratum corneum and look at some of their properties. And here is a simple stress-strain response. So this is basically stressing the skin. You can see here the units are megapascals of stress and the strain, and you can see that the stratum corneum layer is actually remarkably linear. The overall full thickness skin layers are actually quite non-linear, but the stratum corneum is quite linear over fairly large strains and is very, very sensitive to moisture. So hydration of the stratum corneum is critical in terms of its stiffness. If you compare these stiffness values here on the order of, at this humidity, about 70% humidity here, this would be Hong Kong in summer, I guess, at nearly 100% relative humidity. You can see how these numbers change dramatically, but they're all vastly higher than the overall stiffness of full thickness skin, which is shown here over relatively small strains and is only about 0.5 megapascal. In the course of our studies, we've looked at things like the effect of age. We have tissue donors from 29 years old all the way up to well over 88 years in age. And you can see that the slope gradually increases. And in fact, here is a study that we published in 2015, which was the first in this area to actually show what happens as your stratum corneum ages. And you can see here that if you plot the elastic stiffness, how stiff the tissue is, going from donors that were in their 20s or 30s all the way up to donors above 90 years old, there's an increase in stiffness. So we become stiffer as we get older. And in fact, along with the stiffness, if you look at these histological cross-sections, this is the top stratum corneum layer here. This is for a young subject. This is an old subject. We also become flatter. This is partly related to the shininess that we develop as we get older. And I'll say something a little bit more about that, how to treat that in a little bit later. We also, if you measure the adhesion energy of these corneocyte cells, this also increases with age. So basically, as you get older, you get stiffer and tougher. That might not always be good for appearance, but that's basically what happens. So again, coming back to this top epidermal layer and particularly the component of this layer that's the very top, the stratum corneum, things that are critical when you're thinking about cosmetic formulations involve, for example, the sebum layer that's expressed on the top of your skin. This is formed from a number of complex squalenes and lipids and fatty acids. And you can see their relative proportion over here. And this expression is very different in males and females. The lipids encase the corneocyte cells, which are shown over here. And these have a combination of, it's a highly keratinized cell, essentially. No nucleus, because through the desquamation process and the motion through the epidermal layers, it no longer contains a nucleus, although there's very important enzymatic reactions that degrade the cornea-desmosome connections that allow these to desquamate. And a highly corneified, protein-rich cell membrane on the outside of the cell. So when you look at treatments, the kind of innovations that cosmetics companies try to do is, first of all, understand the fundamental interaction of their formulation with the stratum corneum. That's one of the key initial aspects. And what I'm going to show you is a different view of this than what was traditionally taken by the cosmetics industry. Basal stem cell regeneration, of course, is really important. The basement membrane, this is the connection between the epidermal layer and the dermis, is important along with epidermal health. And then, particularly for aging, the collagen and elastin fiber networks in the underlying dermal layer are important. So I can't talk about all of this here, but one of the important things when you look at a phenomenon like wrinkling that occurs in skin is that you need to understand carefully the remodeling timescales associated with collagen and elastin fiber networks, because these are the mechanical components that basically form and stabilize instabilities like wrinkles. So the work we've done over many years has been to use a range of different, what you might think of as thin film techniques, to characterize the top stratum corneum in particular in detail. So we use the usual kind of spectroscopies. Of course, this is not new to the cosmetics area. Most cosmetic companies will have labs in which they do various types of spectroscopy. So we do the same thing here, where we look at very carefully both FTIR as well as Raman spectroscopy to characterize the components, the lipid components, the protein components, the water levels, which all can be quantified. We're also interested, when it comes to treatments, in the penetration of those treatments into the stratum corneum. Now, this is a very interesting area because in the cosmetics industry, the last thing you ever want to do is make a claim that your product is penetrating too deep into skin, because then you're going to get FDA oversight and you're no longer just a cosmetic product. It now becomes a medical treatment, and so the regulations are very different. However, there's virtually nothing that you can put on your skin that doesn't diffuse into the skin, including fully occlusive treatments like petrolatum, Vaseline. These are often thought of as purely topical treatments, but there's low molecular weight components that you can show that diffuse into the skin. One of the particular innovations that we've made in this area has been to understand the water loss that occurs through the stratum corneum layer. And the effect of this water loss is that there's a volume loss in the stratum corneum, and it would normally want to contract. However, it's attached to the underlying epidermal and dermis, and so this contraction is resisted by those layers. And as a result, mechanical stresses, in-plane mechanical stresses, develop in the stratum corneum. So early on in our work, we were interested in what these stresses were and how we could characterize them. So we have developed a method that was basically a technique that is used in thin film material science, where we mount the isolated stratum corneum onto a flat substrate. This is usually either a square glass substrate or a cantilever beam. And we then flow over the top of this a controlled relative humidity air. And this causes water loss, dehydration of the stratum corneum. And as the stratum corneum attempts to contract, it imposes stresses on this underlying glass layer, and that causes the glass layer to deflect. So if you can measure this deflection here, you can very accurately measure the stresses that are developing in the stratum corneum. And so we did that, and what we found, this is some of our early data here, is that we could quantify these stresses, here it is in megapascals, as a function of the drying time. So this is drying here over about 8 to 12 hours in 45% relative humidity, 15% and 5% relative humidity air. And what you see clearly is that the stresses increase. And in fact, the data is the square points over here, and these lines are actually predictions of diffusional models that we've developed. So as water is lost from the stratum corneum, there's lots of intricate details about what the diffusion paths are and how this happens. The stratum corneum wants to contract, it loses some thickness, it attempts to contract in the in-plane direction, and these stresses develop. And the thing that was shocking to us and really surprising was the magnitude of these stresses. So in a dry environment, the stresses can be elevated to 4 megapascals, 4 million pascals. What's a pascal? Does anybody know what a pascal is? Come on, guys. It's one, I know you know this, it's 1 Newton over a square meter, right? It's a pascal. So what's a Newton? Anybody know what a Newton is? It's an apple. A medium-sized apple is 1 Newton. So you take 1 apple and put it on 1 square meter, and you have a pascal. Come on, it's the first time you really know what a pascal is, right? That's a pascal. This is 4 million apples on 1 square meter. That's a lot of apples. So these stresses are very, very high. In fact, this stress here is about maybe a quarter of the way to the yield strength of copper metal, all in the stratum corneum layer without failing. So these stresses are very large. We put a lot of effort into both characterizing these stresses. We have multiple systems now where these cantilevers are all supported with dry air chambers where we can bleed in air at very controlled temperatures and humidities and characterize these stresses. And we've developed models, very precise models that can accurately tell you if you expose your skin to certain environments, we can predict accurately what the stresses will be in your skin. And that model is shown over here. And I'm also showing you this so that you know we know how to use equations at Stanford. This can be done. Now the next thing that you can do here with this technique is you can dry the skin as shown over here. If you then rehydrate the specimen, so we turn the gas supply back to 100% relative humidity, these stresses are fully reversible as water comes back into the stratum corneum. It's absorbed into the stratum corneum, the stresses go back to 0. Now you can apply a treatment, some cosmetic formulation, and you can re-dry through the same process, and what you see here is that the cosmetic treatment has changed the water loss. Less water is lost. If you're using a humectant like glycerin in the treatment, then this would actually bind to water, diffuse into the stratum corneum and hold water in the stratum corneum. And so you see a stress decrease. What's more, if you take different cosmetic formulations, and you can do this with individual molecules, you can look at emollient molecules, you can look at humectants, you can look at surfactants, you can put them together in formulations because they behave differently, and all these stress reductions will be different and quantifiable. On the other hand, if you use a cleansing product, the stresses are elevated because you've basically stripped some of the lipids out of the stratum corneum. You've maybe even created some damage, depending on how harsh the cleanser is, and the stresses actually go up, and they go up by different amounts depending on the product. So these things can be quantified. And what we were, of course, you can use all of this work to determine the efficacy of treatments, the harshness of cleansers, and all of that work can be done. But over the last number of years, we've been interested in what does this mean for sensory perception? When your skin feels tight, what's the mechanism by which this happens? If you put a cosmetic treatment on that feels good, where does that feeling come from in terms of the tightness of your skin, maybe during the duration of a day? And so what is the mechanism by which effects on this top layer are transmitted down to the mechanoreceptors or the free nerve endings in the skin? So this distance here between the stratum corneum and where these first mechanoreceptors appear is roughly 100 microns, maybe 200 microns. And there's no biochemical signaling. So there must be some other mechanism by which this information is being transmitted. So we became very interested in the mechanoreceptors themselves and the type of afferent neurons that they were connected to that would provide signaling to the brain. Now, again, this is a big subject. I actually know that there's a neurology community here, so I'm probably getting myself into trouble. But the mechanoreceptors themselves are listed over here. They're sensitive to different strains. I'm not going to say stresses because it's really the strain that they're more sensitive to. And some of them are sensitive to quickly changing strains. Others are more sensitive to long-term strains and will continue firing through an innervating neuron. You know, the neurons themselves have special function, but what we're mostly interested in is slowly adapting neurons, because when your skin feels tight, it feels tight over a long period of time. And so we're not dealing with short-term perception here, we're dealing with a mechanism that has to involve long-term. And so what we've been most interested in, for a number of reasons, are Merkel cells, which are also the first cells which begin to appear under the surface of the skin and slowly, that are connected to slowly adapting neurons. And so this part of our work then started to become interested in is how do we model the effect of local strains on the firing of mechanoreceptors. Now there's work in the literature, and there's some early work. This is work by Phillips and Johnson, 1981, some really beautiful studies where they took a monkey finger, they located the presence of specific Merkel mechanoreceptors, and then they used an indenter that looked something like this, where they could press this comb down onto the finger, or the primate finger. And they then could measure, by inserting instruments to actually measure the firing rates, the blue data shown over here. So you can see that as different parts of this substrate were imposed, or here it is shown over here in a simulation that we undertook, the firing rate could be measured, and so that corresponded to different strains. Now this is older work. We actually took the experimental data that they had measured, and we used a full three-dimensional finite element formulation to compute, if you were to take this instrument as they did, exactly the same dimensions as they reported, locate the Merkel cell, we could calculate the strains in detail that existed at this Merkel cell. And we then developed a model for the firing of this mechanoreceptor. And our model, after being tuned, shown in red over here, and the experimental results are shown in blue. And you'll notice that we actually capture some features over here, which in discussions with neurologists, they've actually told us that we're probably more accurate than what the experiments were, because the experiments are kind of averaging over the strains that were imposed. We're able to actually have higher fidelity and actually show that when specific features go over these mechanoreceptors, we can pick up those differences. So we were able to do that and come up with an accurate neurological model for the firing of these Merkel cells. So the question then was, well, how do we use this? So we can measure in experiments the stresses that develop in the stratum corneum with different treatments or cleansing products. We have a neurological firing rate model, which if you tell me the strain at the location of this mechanoreceptor, I'll tell you what the firing rate is. How do we apply that? So here we used some more, these were 2D finite element calculations. And this shows a simulation of human skin. This is the top stratum corneum layer. These colors here represent strains. And here what we did was put the stratum corneum on the top, and we allowed it to contract by exactly the amount that we can measure. And if you look underneath, what you find is this big blue region here, which is 0 strain. And that's a classic thin film result. If you have a stressed film on a flat stressed film on a substrate, there will be no strains transmitted to the underlying layer. The only way that you can transmit strains is if you introduce topography. So here's the same model where we've allowed the top surface to actually become deformed, and now you can see through these colors here that strains are radiating down towards the mechanoreceptors. So for activation of mechanoreceptors, in order to have a sensation of skin tightness, for example, you have to have two things. You have to have a stratum corneum that's contracted or expanded, and you have to have topography. Those are the two requirements that you need in order to get strain distributions down to these bottom layers. Well, the next thing is to actually go and take real histological cross-sections of human skin with the actual more precise location of different mechanoreceptors, turn that into a finite element model, and actually compute. Here you can see we've allowed the top stratum corneum layer to undergo the strains that we measure, and here are the resulting strains at these mechanoreceptor sites. So we can take those two things, the experiments, the neurological model, the actual simulations of what actual histological cross-sections would see in terms of strains, to predict what the firing rates would be. So the next part of what we were doing, and this is work we did with collaborators at L'Oreal, was to take 9 different moisturizing formulations and 6 different cleansing formulations and actually test them on subjects. So this assessment was done on 2,000 women in the spring of 2015 and 2016 in France. The cleansers were actually tested on 720 women in the spring of 2017 in China. They all had apps where they could report on their sensation of tightness. These are not expert folks. These are just normal consumers that were asked about their sensation of their skin following the use of these different treatments and cleansers. And we conducted testing on the same cleansers and moisturizing products in California on American skin. So we've got French skin, Chinese skin, and American skin, 3 different places. And the question is, could we simulate what these sensations were? And here is the absolute remarkable result. I remember vividly when we were doing this work that I never ever expected to see such a strong correlation. What we've plotted here is the predicted firing rate from our model based on experimental results measured in California. What we're predicting here, what we plotted here, is the tightness perception score reported by these 2,000 French women and 720 Chinese women. These are the moisturizing products over here. Here, the cleansing products are over here, and what you find is that this all lies along a single line. We can accurately say through experiments and resulting neurological firing rates what the perception that consumers would report for the application of this treatment. This was a model conducted on skin, on cheek skin. We conducted the same studies on forehead and abdominal tissue, and again, with high confidence in all of these cases, we're able to predict this effect. So it was a very exciting study, we thought, and really being able to be so quantitative about this mechanism. And we've subsequently repeated this work in several new studies with actually different collaborators. So here's some work we did where we actually, here you can see the effect of these treatments on drying stresses that we can measure. If you use a cleansing product, you see an increase in stress. Again, put that through the models that we have, and you predict that you would report a feeling of tightness, and in fact, that's exactly what consumers reported here. It's the scatter band here is a little bit bigger scatter band, but still right up here on the curve that we predicted. Now, following this cleanser, you follow directly the cleanser with a moisturizing product, basically trying to repair the damage. And what happens now is this is the same skin, it's been rehydrated, and now the cleanser has been used. The stresses go down, and we predict it should be down over there. And again, what we're predicting is exactly what consumers are reporting. So we're very excited about this ability because it's giving us a quantitative way to actually map the effects of treatments. And of course, it doesn't just have to be these kind of treatments. It can be many other effects of UV exposure, for example, that we could actually predict for consumers. So all of this is based on numerical models of human skin, and here's just a picture showing this kind of model. But a lot of our work has been trying to innovate on the computational models that we have. So where do we get the data? Well, of course, you can take histological cross-sections, but then you only have one plane through the thickness of skin. You can take a replica of the skin surface and image that, or you can use digital microscopy to actually image the top surface of skin. But those you could generate a solid model. The trouble with this is that you only have top information. You have to make assumptions about what might be going on underneath. So a new technique that we've been working with is optical coherence tomography, where we can do in vivo measurements and basically make slices through the skin to get a 3D model of human skin. And so that's shown over here. Here's the result. This image over here are 1,000 histological cross-sections that are actually taken through the skin in vivo and in different locations. That can be then rendered into a 3D model, and this shows a model, a computational model. This is the underlying layer, and this is the top surface here of the epidermis. This is very exciting because we now know the full thickness skin, and our big interest at the moment is the exact location of mechanoreceptors and how they're distributed in these models. So all of this has allowed us to build what I think is the most accurate and high fidelity model of human skin in different locations with a very large number of nodes that allow us to do these computations very accurately. And in fact, when we compare our older 2D models with the predictions from these full three-dimensional models, we're still finding that we have a very good correspondence. So this has sort of allowed us to develop this overall capability of doing experiments in the lab that are relatively fast to do, of different treatments, and then we can actually predict what subjects would experience in terms of perception. Okay, so let me show you in the time that remains a few other areas that we've used these studies. One has been on looking at the role of different molecular components in formulations. And in this particular study, we've worked with BASF, who are purveyors of course, many different chemistries, and in this case, many different emollient molecules, which are all shown over here. And these are used in different cosmetic products for different reasons. They all have different molecular structure, they have different functionality in terms of their functional groups, they have different molecular weights, they have different polar surface areas, and they are all effective at decreasing the rate at which the stratum corneum dries. And again, you can see here are measurements from these drying stress curves. Here again is the control. You can see using different individual emollient molecules here, how each one of them has a different effect. And in fact, this is again something, I'm not going to be talking about it here, that we can model quite accurately. The solid curves here are actually our model predictions, which now include the loss of water and the uptake of the emollient, all of which are taken together change the volume of the skin. And so this is work that we published a couple of years ago. In fact, this work here won a very significant prize in the International Federation of the Society of Cosmetic Chemists. There was a meeting in Paris that my student Chris Berkey actually received an award for this work. And here what he's showing is that if you take individual emollient molecules, then as we expected, if you also measure their penetration volume, how much of them penetrates into the stratum corneum, this essentially involves partitioning into the intercellular lipid boundaries, then that scales linearly with the reduction in the drying stresses that develop in the skin. And that can be modeled. Now, if you put the same emollients into a formulation, then you see some very interesting effects. Now, this is a big topic here, which I won't cover here at all. The effect of individual molecules and their efficacy in a treatment is modified when you put them in a formulation, as you would expect. There's synergistic effects between other components, the emulsifiers, the stabilizers, the surfactants that might be present in a full formulation. But you can see in this work, all of these molecules here were put into a full formulation, and you can see how the curve is shifted in this direction. So essentially, in a full formulation, for all sorts of biochemical reasons, you get greater penetration of these emollient molecules into the stratum corneum. So the penetration increases. The stresses move up a little bit, but not that significantly. Now, another component that is often present for full formulation is a polymer addition that's added to stabilize the formulation. It's kind of a cool topic that we're looking at the moment. Very stable formulations with stable micellular droplets penetrate much more effectively into the stratum corneum than the individual components. Well, that's not expected, right? Because energetically you have to break down this micellular barrier that introduces another energy barrier to actually the uptake of the molecule. But nevertheless, this happens. And so these polymer additions are made to stabilize these emulsions. But as the formulation begins to dry, it loses water, these polymers often form a residual film on the top surface of the stratum corneum. And these top films that form, as these polymers basically form a film, they might cross-link, depending on if they're a polysaccharide, for example. They might actually actively cross-link, or they might just entangle. But they themselves develop stresses. So you have a stratum corneum layer, which we've been talking a lot about the stresses that develop. Now you've got this treatment sitting on top that has a thin film that forms. You can't see the film. It's very thin, on the order of several microns to maybe 10 microns, and yet it's forming a film as well. And it turns out that those can be used not just to stabilize the formulation, but as anti-aging treatments in the following way. Instant wrinkle reduction that feels good. Let me show you how this works. Here are two images of the same subject, where here you see the appearance of what would be called fine lines and crow's feet next to the eyes. And here's the same subject after the application of one of these anti-wrinkle formulations. So what the heck is going on? Why is it that you can essentially so quickly reduce wrinkles? Well, one of the most effective ways to do this is to use a treatment that forms a very tight film on the surface of the stratum corneum. And here's a product, Plexiderm. You can buy it on Amazon, and you apply it to your skin. It forms a very highly stressed layer. Here we've measured the stress. There's a 44% increase in the stress on the stratum corneum because of the presence of this film. And what that does is two things. It actually flattens, whoops, hang on, I'm going the wrong way here. It flattens larger wrinkles, but more importantly, it causes micro-buckling. So it actually causes, at smaller length scales, it causes little instabilities that cause these micro roughening of the surface. Now, if you remember back to that early slide I showed you earlier on between young skin and old skin, young skin is textured. There's lots of very small wrinkles on the skin of young people. Their skin is more tight, but the top surface is topography. As you get older, your skin gets flatter, smoother, and you become shiny. So it's actually this micro topography that's so important. And what these treatments do is two things. One is that it helps to flatten the skin at larger length scales, but it causes this micro-texturing, this instability, it's buckling instability basically, at low length scales. So we were interested in developing something better here. And here we worked with a company called Rodin and Fields in San Francisco to develop a formulation that would involve both a moisturizing product, some of the components are shown here, plus a film tensing component. This is basically xanthan gum. So if you just take the treatment with no moisturizing components, you see you get this increase in stress. If you take a full formulation with moisturizing treatments, you get a stress that looks like this. And if you have no film former in here, then you get a reduction in stress. So we wanted some moisturizing, but we also wanted some stress. And what we did in this work was to do some optical imaging. This is, again, a human skin in which these treatments were applied, digital imaging, and then you take a power spectrum of the resulting surface, and that's shown here. So this is the normalized power plotted on the vertical axis. This is the length scale on the horizontal axis. So this is basically the size of wrinkles. And what you see is that before application, there is this bump over here. This represents larger scale wrinkles on the order of 100 to 1,000 microns. But with the application of this treatment, you can see that there's flattening of these large wrinkles, but this is the important thing here. There's, at the length scales of 10 to 100 microns, there's an increase in surface topography. The way I like to think about this sometimes, imagine a flat lake of water, no wind, it's completely flat. You take a rock and throw it in, and you see the rock land, and it creates a big wave that will radiate out from where it left. Now, if the wind is blowing and you've got a lot of small ripples on the lake, do exactly the same thing, throw the rock in, exactly the same large waves will form, but you don't see them because they're hidden by the microtexture that's created by the small waves. So this was a very successful approach, and in fact, I won't go through all of the sensorial aspects to this, but participants in the study of this work reported that not only did they have, in their opinion, a better appearing skin, but they didn't have this tight feeling that you would get from some of the treatments that just generate stresses. Okay, last little topic to talk about here, and then I will end. This is work we're doing on the difference between male and female skin. So this is actually my student, Ashley. This is her here. This is her here if she was a guy, I guess. And not just identify as a guy, but actually be a guy. What are the differences? Now, surprisingly, in the literature, there's very little information, particularly when you get to the level of the stratum corneum, on the effect of different sexes on skin. We know that there's more sebum expression in males, so we know some basic features, but other aspects were simply not known. So, of course, much of this is hormone-driven. Estradiol, in the case of females, inhibits sebum production. Testosterone, in the case of males, promotes sebum production. Male skin is thicker. It's about 20% thicker. It's thought to be mechanically stiffer. There's evidence of that. That's predicted in the literature. And there's some confusing evidence about whether there might be lower water diffusion rates. So the diffusion of moisture through the stratum corneum is less. The opposite for females. So this is something that we've embarked to study further. And again, coming back to our stress measurements here, the first thing that we found, and this was really surprising to us, which has now been reported on multiple donors, many different specimens, very low amounts of scatter, here is the typical dehydration stresses that develop in female skin, and here are under exactly the same conditions, the stresses that develop in male skin. So male skin dries much more slowly than female skin. Water loss through male skin is much lower than it is in female skin, and the stresses that develop in female skin is much higher than what it is in male. Now, this is very much of an ongoing study that we have at the moment, where we're using spectroscopy like this ATR, FTIR, where we mount stratum corneum directly onto an ATR crystal. An evanescent field is formed as we shine infrared light through the crystal, and we can then image close to this bottom surface levels of water, for example. We can track water peaks in the absorption spectra of the FTIR. We can also look at other effects of lipids and lipid ordering. We can see how that changes with water loss. We can look at hydrogen bonding, which affects protein structure, and several other aspects. And you can see here again in FTIR work, so it's completely separate measurements now. Again, water diffusivity that we measured with this spectroscopy in male tissue is much lower than it is in female. So this is a remarkable difference, and something that we're wanting to understand, well, where does this come from? So one aspect that we've been looking closely at is lipid packing and lipid ordering, because these are the main barriers for diffusion. And we're starting to see that there are some differences in male and female that tell us that female lipids might be more disordered than they are in males. In addition, we've been doing a lot of X-ray scattering results, studies. This is just normal X-ray, low-angle X-ray scattering that we are doing on systems that we have on campus. You can see that it's really difficult to distinguish some of the peaks here that are related to the lipid structures. They're very characteristic peaks that form. And so this is the area where we've gone to the linear accelerator at Stanford, and we're doing low-angle wax and SAX studies. So this allows us to interrogate structures, in the SAX area, small angle X-ray scattering on the order of, tens of nanometers, and then we're in wax down to even smaller length scales. And here we just in fact completed a number of days on the accelerator, and you can see the spectra that we can measure now is much more clearly defined. And we have some really cool results here that are confirming our understanding that there's more ordering in the case of female skin than male skin. One other aspect here. So when we do all of the kind of studies that I've talked about, and we now look at a range of different moisturizing formulations, those formulations are always more efficacious in males, here as measured in how much they reduce the dehydration rates and the stresses compared to females. So yellow is female, blue is male, and you can see in nearly all cases except this one over here that there's a bigger effect of male skin. So there's a bigger effect. Okay, that's cool. We can now do all of our computational neurological modeling, and when we do that and we follow it with clinical studies involving subjects, we get, again, as we have in many studies, the same results that we saw before for females. But we don't get the same results for males. Our neurological model and our experiments predict similar effects, so we're seeing the same effect. But when you ask men, how does your skin feel, you get a different answer. And we're very interested in why that is. We think part of it is simply that women subjects are more attuned to their skin. They've spent a lifetime of caring for their skin. They can tell you how their skin feels. Whereas guys tend to say, my skin feels fine. And that's what we're seeing here. We know there's a difference because we can measure it on male skin, as we have in this work here. The difference, the effects are the same, but men simply don't tell you that. So it's a very interesting aspect. We're retuning our questionnaires and how we actually ask subjects to see whether this is really just a question of men in general, because I'm talking on average here, are simply not as aware and maybe even be tuned to say, I feel fine. I don't have any difference in feeling. Okay, so with that, I'm going to end. These are areas that we've been working in on human skin. I couldn't go into full details in any of these. I'm happy to answer any questions, or if you want to email me afterwards, please do, and I'd be happy to talk to you about it. Thanks again for inviting me.
Well, it's really interesting, inspirational talks. I'm sure they have many questions from the audience. Yes.
Okay, Professor, thanks for your wonderful presentation. And I have 3 questions to ask you. As you mentioned, the human skin's characteristics, such as the thickness and the elastic and the sensitive, are different. So, how about the broad applicability for this research result? And the second question is about the biomaterial will promote the wood healing of human skin, and where it will inhibiting the regeneration of nerve ending, and how to address this problem. And the third question is...
Hang on. I need to understand the first question.
Okay.
So what was the first question you asked?
The human skins, such as the thickness and the sensitive and the elastic, are different.
Right.
So how about the broad applicability?
Well, so I mean, I think that's what we're trying to do in this last work that I was showing you is how does this play out in male skin? So we know that when we look at male skin in general, the water kinetics are very different. This probably relates to slight differences in the lipid structure. That would be enough to actually change the water kinetics, and that's what we're looking at. But other than that, the basic models and what we find with different formulations is very similar. The effects typically are a little bit larger in male skin because we get a bigger effect. Now, I should add, this is studies that are underway. One thing that we're interested in is the level of sebum in males is higher. And the sebum, it will be dissolved in formulations. So one of the things that happens in skin, male skin that has a higher level of sebum, is that when you apply a formulation, you automatically get a higher loading of components that are now available to diffuse back into the stratum corneum. So there could be effects like that as well. But I think other than that, the models, we're seeing the same thing in male skin and female skin. When we make predictions of the strain distributions and the firing rates of afferent neurons, we get similar results from the models that we have. So, I think in that area there is broad applicability of this work for both male and female. It's just in terms of sensorial perception, that's what we need to understand more, is why men are not reporting the same perception as women are reporting.
Okay, the second question is about the biomaterial will promote the wood healing of human skin while inhibiting the regenerative of the nerve ending. So how to address this problem?
Oh gosh, I mean, no, that's not something that we've looked at. Nerve regeneration is not. I mean, the work we've done in wound healing has really been focused on hypertrophic scarring. There may be some application of the techniques that we developed for keloid scarring, but that has a much stronger genetic component to it than hypertrophic scarring. We've not, in that work, we've not looked at what effects there might be on neurological regeneration, if that's the question that you're asking.
The third question is about sorry. How about the mechanism of the human skin for anti-hydration and anti-swelling? And is there any inspired for artificial skin?
Well, I mean, our work has been really centered on human skin. Well, all of the work is done on actual human samples. We've not worked on artificial skin. There are various skin analogs out there. They're not appropriate for the kind of studies that we've been doing. We are interested in cultured skin, and we have some work underway in that area. I didn't mention that here, but these are skin models of real human tissue.
Okay, thank you.
Yeah, back there.
Thank you for the presentation. I wanted to ask about the response of people to the cosmetics. So you talked about how you tested the cosmetic in different places, California, France, and China. And I believe they have different atmospheric environments.
Yeah.
And did you notice any differences on how the environment affect how the skin of these people respond to the cosmetic? And is there any approach to ensure that different people in different environments are going to be able to respond to the cosmetic better compared to others?
Yeah, that's a really good question. In fact, the study we did on the moisturizers was done in the spring on 2 different years, as I indicated. And in fact, it turned out that the first of those years was a very wet year with high levels of hydration, lots of rain, and the next year was very dry. And so the studies were done under these very different conditions. The cleanser studies were done in China in a completely different environment. I didn't mention this when I actually showed the data and how it all fits on a very, very tight line. That makes it even more remarkable that you can take subjects on different continents and have their response in terms of the sensation of tightness being similar and all basically collapsing onto a single curve, which would then also fit with measurements that we'd made in California, in different environment, and taken from subjects that actually were living in the States. So why is that? How can that be where you have cosmetic formulations used in different places in different years with different environmental conditions. Well, the first thing I will say is that cosmetics companies do formulate treatments differently for different environments. So that is done already, okay? That was not part of our study. The treatments were all exactly the same. What I think it's showing is sort of what you see in the results. It's the delta that matters. It's, you could be measuring the effects of perception in a very wet year where your skin might be more hydrated. But when you use a treatment and you look at what happens during the course of a day, because our predictions are at the end of 8 hours, the delta, the effect of the moisturizing product is the same no matter whether you're in a year that was very moist or a year that was much more dry. So I think it's the delta that triggers it. And there's probably some really complex underlying neurological aspects to those kind of studies, some of which we're working with neurologists to try and understand better, but it becomes very complex because there's all sorts of habituation effects that occur with neurological responses. And anyway, I think the main answer to your question is that when you use a treatment, it's the delta that matters, and that's what we saw in the data that we measured.
Okay, one last question, gentlemen.
Hello, Professor. Thank you for your interesting presentation. I have one question. So I believe that oil skin and dry skin, they may react differently to the same moisture or the cleanser. So how will you differentiate them in your research or when applying the moisture?
Yeah.
Thank you.
Especially on the consumer side, we often do use subjects that have been pre-screened. So, we might have different buckets of subjects. Some subjects might have, more oily skin. Other subjects might have drier skin. Some people might have very sensitive skins. The work that I showed here did not include that. This was just 2,000 people that were basically selected. In other studies that we've done, we are now becoming a little bit more careful about how, what the starting condition of the skin is and what the subject is reporting. So you're right, that can be important, and it's something that we're doing in current work that we have underway.
Okay, we've overrun the event. I think we have so many questions to tell us how interesting your work is. So, so please join me to thank Professor Oscar for his excellent talk and sharing. And I think he will give another talk in the afternoon on different topic. Yeah. Right. So if you have interest, please join. Same venue, I believe.
Yes.
Yeah. Okay. Good. Thank you all for joining today.
Thank you.
Excellent. Interesting.